Good afternoon, everyone. Welcome to today's science session. I'm Liz Bothwell, and I'll be your moderator today. We're so glad to have you, and I just wanted to share a quick reminder or two before we start. We will have time for questions after the presentation, and you can submit those through the chat function. If you'd like a copy of the presentation, please download those in the handout section. And please look out for a post-event email with a brief survey and a link to your certificate of attendance. Now I'd like to welcome you to EREF Science Session, Transforming Textile Waste into Renewable Energy. And we're really fortunate to learn from an amazing speaker today. You'll meet Dr. Sonja Salmon, an associate professor in the Department of Textile Engineering, Chemistry, and Science at NC State University. She spent more than 20 years working in industrial R&D before starting at the Wilson College of Textiles, mentoring students involved in funded projects. Her research team is engaged in new developments in biotech textiles and sustainable polymers, with a special focus on enzyme fiber interactions that enable increased utilization of bio-based materials for textile applications. Her work on enzyme fiber interaction encompasses enzymatic modification, degradation, and processing of fibers, enzyme catalyzed synthesis of fiber-forming polymers, and enzyme immobilization on fibers for the development and production of a new class of biocatalytic textile materials. So now please take it away, Sonja.
Great. Thank you very much, Liz, for inviting me, and thank you all for attending and listening to the research work we've been doing. I kind of wanted to start this presentation with something pretty familiar. On the picture right there, you see a cotton polyester t-shirt. This is something that most of us have in our closet. We wear them. They're great performing garments. They're comfortable, they're durable, but they do have an end of life. And so one of the issues that faces textiles today is that textiles, being highly engineered, pretty complicated materials, they're not so easy to recycle. They tend to have mixtures of fibers in them, and that makes it difficult to reprocess because those fibers can have very different properties from each other. So given that we have this problem, my team has undertaken to address this textile waste problem. To give you an idea of the scale of the problem, first, if we just consider about global fiber production, that is around 110, 120 million tons of fiber is produced annually. There's a reference at the bottom of the page there to Textile Exchange, a great organization that puts out a free market report every year. So if you're interested in tracking those numbers, I recommend you look at that report. So that report will also give a breakdown of what the composition of those fibers are. And PET, polyethylene terephthalate-based polyester, is by far the largest single textile fiber. It commands 54% of the fiber market. And number two is cotton, which commands 22% of the market. So you can actually see that pretty much 75% of the global textile market is dominated by these two fibers. And polyester is the main polymer in polyester, and cellulose is the main polymer in cotton. Cotton is almost pure cellulose. So we use these materials, but then we throw them away, and essentially most of them go to landfill. The vast majority goes to landfill, about 11 million metric tons in the USA alone, and that's a little harder to get global numbers, but we've made an estimate that the global waste would be 65 to 92 million metric tons. So not only is that a solid waste problem, it's also a lost value opportunity. So there could be value in that waste, and we certainly don't want it to be accumulating at gigaton scale, which is what we are on the path to do. So the challenge is there are actually many types of recycling options that you could consider. What I'm going to talk about today is a kind of a bio-based approach to doing what we call enzymatic fiber separation and recycling of the resulting materials. So we looked at the biodegradability profile of textile fibers, and the literature there is some literature on it. It tends to be fairly varied on what the quality of the results are, and biodegradation studies tend to take a long time. So sometimes the results are a little bit incomplete compared to what the standard methods require. But if we look at this, soil, compost, and seawater, the time for those types of degradation to occur is pretty long. The quickest in this list is anaerobic digestion. So from the perspective of saying, okay, if we have all this solid waste and we want to process it quickly, let's have a look at this method. That would be already the quickest that's kind of on the books. So if you look at the studies of cotton and polyester in anaerobic digestion, cotton does meet the criteria for biodegradability by anaerobic digestion within that 30-day window, and polyester fails that test. It will not biodegrade under anaerobic conditions. So we have two types of materials they behave really different from each other when you expose them to these biological systems. But we can take advantage of that, because if we consider textile waste, of course, there are many different textile wastes, but if we kind of simplify it for the sake of today's discussion, that we're talking about cotton and polyester, we could imagine cutting and shredding this polyester cotton material, subjecting it to an enzymatic treatment where enzymes are biological catalysts. They're protein molecules. There are very many enzymes, thousands of enzymes in nature, and some of them are very specialized to degrading particular substances like the cellulose that's in cotton. So we could get some of these cellulase enzymes and degrade the cotton portion into soluble materials and small fragments that could be fed to some biofuel production, like anaerobic digestion that we were just talking about. And in this process, the chopped up cellulose, which actually is glucose and small fragments of glucose, that is a feedstock for microorganisms to produce biogas, which is composed of methane and CO2, methane and carbon dioxide. So methane is a drop-in fuel, so you could use the methane as a heating source or run buses or what have you. But then we've taken the cotton and we've converted it to something that can turn into a biofuel. And at the same time, the idea is that we've separated that cotton from the polyester fraction, which we could recover, essentially cleaning the polyester for recycling. And then there may be some other co-products and residuals that need to be thought about. So within this project, which was funded by the EREF organization, and we appreciate that very much, we carried out lab scale investigation of these questions. So our goal was to minimize organic waste to landfills, because it's really not doing anybody good sitting there, and it could actually potentially cause problems from methane emissions. And we wanted to see if we could generate new revenue streams, either from biogas generation or from fiber recycling. So we approached this with these 5 objectives: to enzymatically deconstruct the textile waste and deliver the resulting slurries to anaerobic digestion, evaluate the quality of the recovered fibers for recycling potential, evaluate the level of anaerobic digestion conversion of degraded components to biogas, characterizing the chemical byproducts, and generating a preliminary cost analysis for feasibility. Here is a little picture of kind of what a generic experiment that we would carry out would look like. This is showing what a blue dyed 100% cotton fabric would undergo in this experiment. We would start out on the left side with our fabric. We would cut it into small pieces, put it into this 250 milliliter bottle, add these stir bars to it. Notice we actually used 2 stir bars. That was deliberate to give unbalanced mixing. That would be kind of like a pounding action. That mechanical action is really important. And after the time and temperature conditions that you see in the lower right, 19 hours, 50 degrees, pH 5, we would end up with a slurry. So that all of that fabric on the left turns into a slurry shown in the middle. Then we would take that slurry and filter it over a filter. In this case, that was a 1.6 micron filter. And we would collect all of the solid fraction that's remaining. We called those small solids. And what went through the filter would be what we called our glucose syrup, or the weight loss. And we did do HPLC analysis on some of these liquors and confirmed that glucose was the main component in these syrups. You'll notice that in this case, the syrup is blue colored, and that's because the dye that we applied to this fabric is what's called a fiber reactive dye. So it chemically reacts with the cotton and forms a durable bond with the cotton. So the enzyme, when the enzyme is chopping up the cotton, it doesn't necessarily separate the dye from the glucose. The dye may still be attached to the glucose or to the fiber fragments, as you see there, remaining as the small solids. So among the things we were interested in is we're not all wearing white clothes, right? We're not wearing just bleached white material. We are wearing a lot of colors. So we wanted to see to what extent did those dye molecules get in the way of the enzyme actually attacking the polymer. So the top chain would kind of be a schematic of what a bleached cotton glucose polymer would look like. The enzyme could attack between those glucose residues. Whereas when a dye is present, and especially when the dye is actually attached to the glucose, you would cause some interference. The enzyme might just not get to those bonds to break them. And that's in fact what we saw. We structured our experiment to test the undyed 100% cotton versus RB19, that's reactive blue 19. It's a fiber reactive dye that can attach to the cotton cellulose in one position. And we compared that to the reactive red 198, which is a reactive dye that can potentially attach to the cotton in 2 positions, so act like a cross-linking agent. And we can see that the undyed cotton was completely converted to a slurry under the conditions of the experiment. In other words, there were no large solids, none of those original pieces of fabric were still intact. It was either the glucose syrup, the blue part, or the slurry, indicated by the red bar. This was also accomplished for the reactive blue 19. You'll notice that the glucose portion, the weight loss, the glucose syrup, was a little bit lower. So we can see that the dye was a little bit getting in the way, but didn't interfere in this particular type of experiment. On the other hand, the reactive red dye that was capable of cross-linking did still have large solids remaining. So if we were to look at a bottle at the end of that experiment, we would still see in the upper right, you can actually visually see some fragments of fabric still remaining. So this, of course, shows clearly that the nature of the dye gets in the way, and that should be our idea there is that this could help inform textile designers about their choice of dye stuffs, and possibly also dye companies. You might want to start thinking about and investigating how you might change your dye stuffs to be maybe more working better with some of these waste treatment processes. We did find that if we extended the treatment time for a longer period of time, we did get more reaction. And if we did an actual double treatment with fresh enzyme, we could completely convert that red fabric also to a slurry. So this, again, 100% cotton, just testing the impact of different kinds of dyes. Another very common treatment of cotton is to put a durable press finish on it. And durable press is great. It gives you kind of that no iron type of fabric, but the way that's achieved is by putting chemical crosslinks between the polymer chains. And these chemical crosslinks can get in the way of the enzyme. And this was definitely true. We observed this on the far left. You can see that essentially trying to treat a durable press treated cotton with enzyme, we just had pretty much all large solids remaining. So enzyme by itself didn't work. We then investigated, this was a master's student working with me, Jeannie Egan. She looked in the literature and studied some different ways to kind of swell cotton. That would be with a cold alkali treatment. So we tried that next, and it did help a little bit, but it still was not taking care of those durable press crosslinks. So she investigated further and found out that acid could be used to help degrade those durable press crosslinks. And when there was a kind of single acid alkali treatment, it got, quite a bit better. And with this acid alkali treatment with a double enzyme treatment, it worked even better. Subsequent to these results, she did a further investigation of treating with acid only and eliminating the cold alkali. So acid alone with the double enzyme treatment worked very well also. So that was all about cotton, but now we take cotton, there's one more obstacle to get through, and this is when you put cotton together in that blend with polyester. So we did this in several ways. This first example shows just kind of blended fiber. So in this case, the polyester is dyed red with a dispersed dye. Don't get confused about that. We've now moved to a different kind of dye. We've dyed the polyester red. We've left the cotton white. We've put it into our bottles. The one on the left has buffer. The one on the right has enzyme in it. And then we put it through our treatment, and you can actually visually see the white slurry there that results from the cotton fiber being degraded, and the polyester remains intact. There's no red stuff floating around in this particular experiment. In this case, we would use a larger screen to collect that polyester fiber. We would still use our small 1.6 micron filter to remove the cotton fiber fragments, the little tiny slurry stuff. And here we get a clear glucose liquid. And what it looks after it's dry is the undegraded blend on the left there and the clean polyester on the right. So this is a way that we can clean up that polyester, and polyester is well known to be thermally recyclable. You can melt it and re-extrude it back into fiber again. If you can't use the kind of shape that it's in, if it's too tangled or too short, you just melt it and reprocess it. Of course, the cotton is no longer long enough to turn back into a yarn, but these little slurry particles, maybe those are interesting, and so we wanted to investigate what properties those had. So here you can see our investigation of the cotton fiber fragments, where in the top picture, here we dyed the cotton blue, and we degraded away the cotton, and so you can see the white polyester remaining after the enzyme treatment. In the lower microscopy images, you can also see that the smooth polyester fiber remains, but the cotton, that kind of twisted ribbon cotton structure, has been completely degraded to these little fiber fragments. We looked at properties like crystallinity and found that these remaining fragments are highly crystalline, more so than the original cotton, and that the undyed cotton definitely breaks into smaller fragments quicker than the dyed or durable press treated materials. So then we wanted to check whether that polyester that was remaining had similar properties to the starting material. On the left side is infrared analysis, where we just, on the top are kind of two control materials, 100% cotton and 100% polyester, to show you the fingerprint of what those fabrics look like. And we're kind of especially pointing out this region around 3400, which is the hydroxyl stretching band, which would be very prevalent in cotton fabric, but not so strong in a polyester. It only has hydroxyls at the end group, so it's not a strong signal. And so you can see that before the treatment on the left, the 50-50 poly cotton fabric has characteristics of both types of fabric, but on the right, the residual polyester has much more the fingerprint of the pure polyester fabric. And this was also borne out by looking at the melting properties of polyester. Our polyester reference had a melting peak at 254. That was also detected in the polycotton blend, and then that 254 melting peak was also present in the residual polyester. Cotton, of course, does not melt, so it does not have a melting peak. We did find, and I want to emphasize, that efficient washing was needed to remove trace fragments of those degraded cotton fibers. So that's part of the processing conditions that would need to be paid attention to. Excuse me. So here we move into our objective 3, which is to carry out anaerobic digestion of the textile slurries. Along the x-axis, you see our untreated, so the white cotton, or sorry, the undyed cotton you see is the undyed cotton on the left, the red cotton is in the middle, the blue cotton is on the right, and 1x, 2x, and 4x are different enzyme doses that we applied to these fabrics. So you can see that as we increase the enzyme dose, the chemical oxygen demand, or essentially the release of glucose into the slurry, was higher, and this happened in all cases. And this presence of chemical oxygen demand is a good indicator that the slurries would be a useful feedstock for anaerobic digestion. So then these experiments were actually carried out in small scale. Our collaborator, Doug Cal, in environmental engineering, has this little kind of mini anaerobic digester setup where he's tracking the flow of gas evolved from these little digester vials using a respirometer, and then collecting the resulting gas in gas bags that can be subsequently analyzed by gas chromatography. So here are the summary of the findings. On the left, you see the background controls in the green bars. So when there was no slurry added, the volume of biogas was kind of just baseline. You know, there's a little, these are sludges. These sludges were actually taken from an active anaerobic digester at a local municipal waste treatment facility. So that would just be kind of the background biogas generation. And then we had a positive control where a defined amount of glucose was added. That's the purple bar. And then the other ones, again, the yellow bars are the undyed cotton, the red bars are the red dyed cotton, and the blue cotton. And so basically we see that in all cases, we were able to get methane generated. It varied quite a bit. These are they're quite variable experiments because you are dealing with kind of a sludge from an anaerobic digester, so the microbial community in there, and handling that causes some variability in the experiment. But generally speaking, we found a methane yield in the biogas of around 40%. This is a little bit lower than I think what you would get in a commercial anaerobic digester. Usually, I think 60% is the normal amount, but we did get gas generation. In our objective 4, we wanted to know what happened to those dyes that were present. Again, remember that original picture where I pointed out kind of the blue glucose, the clarified blue glucose liquid. So there's dye in there, and we wanted to know what was there and whether it would have an impact. In the prior slide, since you saw methane, kind of good methane yield in all the treatments, the level of dyes did not seem to be having a very negative effect on the performance of the digesters. Here, this was another collaborator, Nelson Vinuesa, and his student Zoe Milburn. She analyzed the soluble dye products using liquid chromatography with high resolution mass spectroscopy, looking for what type of components are present. This is a pretty involved type of analysis. The mixtures are very complicated. So this is just a kind of a subset of the data. But here gives you a little bit of an idea. The ranges of color components that were released into the solution was in the tens to hundreds of parts per million of compounds, of colored compounds. And on the table on the right, you can see that this table kind of highlights that there are a little bit of difference between what you detect in the liquid after enzymatic digestion versus what you detect in the liquid after the anaerobic digestion has occurred. At the bottom, you see 2 example structures, and that shows that we did confirm that the dye molecules were attached to some of these glucose residues from the polymer. So with that information in hand, we carried out our 5th and final objective, which was the preliminary process feasibility. To do this, we considered 4 cases. Case 1 in the upper left is 100% cotton, where we just imagine cutting and shredding the cotton, subjecting it to the enzyme hydrolysis, and feeding the entire slurry to anaerobic digestion to get biogas. So case 1 is about, does it make sense to take 100% cotton, chop it up, and send it to anaerobic digestion to make methane? Case 2 is taking the 100% cotton, but in this case, there's a filtration step where you remove those cotton fiber fragments, the little pieces of non-degraded cotton, remove those, and just send the clarified syrup to anaerobic digestion. The reason for that is the idea that these cotton fiber fragments could actually have some value and be useful. Case 3 was the polyester cotton case, where you would separately remove the polyester fibers in that first filtration, the coarse filtration fraction, and the cotton fiber fragments in the fine filtration fraction, and then send the rest to anaerobic digestion. And case 4 is kind of a special case of case 3, where you might have a higher value fiber than polyester, where your kind of motivation for doing this might be less about the solid waste aspect, but more about actually recovering the fiber itself for a fiber recycling program. So the numbers shown here, I want to put a big caveat on these. These are very rough numbers. Capital and labor costs were not determined in any of this assessment, nor were any renewable fuel incentives or environmental incentives considered. The rationale for getting these numbers is actually detailed in our report, which is publicly available, and it will be shown in a link on the last slide. So let me just highlight a few things of our findings. So first of all, the top row is kind of the same for all the cases. We said, well, you would save some money by not having to pay a tipping fee to get rid of this waste. So that's the plus 66. That's kind of the average tipping fee, US dollars per ton in the United States currently. But then, of course, you'd have to pay something to carry out that enzymatic fiber separation. And we have roughly estimated that at this $140 per ton. And then you start to look at the differences between the treatments. So there's the value of recovered synthetic fiber, of course, is not relevant for cases 1 and 2 because they're 100% cotton. They have a different value between case 3 and 4, with the idea that the specialty fiber in case 4 has higher value. And then there is the potential value of the cotton fiber fragments. Now, this could be very debatable. We don't know exactly yet, but actually we're continuing the work. We're very interested in these cotton fiber fragments and are continuing research on them. If you look on, say, a generic website like Alibaba and look for microcrystalline cellulose, that actually has a value. Microcrystalline cellulose is used in a variety of products. And so if you take the nominal value of microcrystalline cellulose at a per ton scale, you could get a potential value out of that fraction. What we struggled with was actually giving a positive value to the generated biogas itself. Here in the United States, methane is pretty cheap. So really the best we could do was kind of guesstimate that the process cost of carrying out the anaerobic digestion would offset the value created by producing the methane. So that was kind of like a cost neutral element of this whole process. And then the bottom line is that we found that cases 2, 3, and 4 had kind of the most compelling story, where interestingly, the case two, if we really give a significant value to those microcrystalline cellulose fragments, could potentially have a high value. We did a lot of measurements. We took a lot of metrics in this study. This is a little bit too intense maybe to go through in detail, but I just wanted to highlight that this table is summarized in our report. There's a lot more information in our final report, so if you have interest in this, please do refer to the report. I think maybe to draw your attention to the lower right, the last few lines there, we did measure biogas production, kind of an average of 40% over our lab scale trials. Interestingly, we also measured some hydrogen production. So that would be maybe a new angle on looking at this. If hydrogen is desired as the gaseous product, then you could perhaps choose microorganisms that would be more biased towards making hydrogen. And then the estimated enzymatic treatment process costs. I don't want to advertise too strongly. This is kind of an estimated thing. This is kind of an estimated value. It's not really a maximum value. It's, I think, maybe a practical value if you're trying to do some cost assessments up front. I think that would be maybe a reasonable number to use. And then the potential value, I mean, in our calculations, we estimated up to about $540 per ton, but it could be lower and maybe it could even be higher. So we demonstrated this integrated process in laboratory testing. We did show that there is recycle value if we can recover the synthetic fibers and the fragments that could help the economic feasibility of doing waste treatment in this way. And we found the concept was most applicable to the 100% cotton or cotton blends with one other synthetic fiber. If you have multiple synthetic fibers, this treatment, of course, would not be able to separate those fibers from each other. In addition to cotton, I should mention, this process would also work for other cellulosics like rayon and lyocell, and even some what they call bast fibers like linen and ramie and hemp. So the future work that we would recommend is to scale up from grams to kilograms in a type of equipment like a belly washer. This is a type of machinery that's actually already used in the textile industry for processing blue jeans, for example. Trying this tests in larger scale anaerobic digestion to essentially enhance the methane generation potential by having larger systems, and then collaborating with folks who are interested to identify some field test opportunities and further explore the value creation from the degraded cotton fiber fragments, which, as I mentioned, is something that we are continuing to do. So with that, I really want to give my thanks and appreciation to the project team, my co-PIs, Doug Cal and Nelson Benueza, and then the graduate students working on the project, Jeannie Egan and Zoe Milburn, and Chloe Andreasen was an undergraduate student working on the project in Doug's team. This project benefited actually from a fair amount of work that even preceded the project, so there were a lot of others who have helped this along, and thanks to them. And then also really want to thank EREF for supporting our work with funding. Thanks and a shout out to Kanika for providing some supplemental funding to our work, and thanks to Novozymes for providing enzymes to the project. The QR code will take you to my textile biocatalysis research team website, where there is a publications tab where you can find the final report, or you can go directly to this link here that EREF has provided. So the final report is there, as well as publications that we made from this project, as well as Jeannie Egan's master's thesis is cited there, where you would find additional information about this work. So with that, I thank you very much for your attention, and I will be happy to address questions.
Thank you so much, Sonja. That was amazing. I know I learned a lot, and I hope everyone else did too. Let's get right into the questions because we have a few. How might textiles in AD affect the maintenance and operation of the digester?
Great question. Thank you. Actually, a main driver for us doing this project was the fact that we could turn the textiles into a slurry. And of course, note that what we're doing is we're removing anything that wasn't converted into a slurry is removed by filtration before that slurry would go to the anaerobic digester. So my guess would be the person asking the question is probably very familiar with anaerobic digesters. Maybe others in the audience are less so. But these digesters, they have big mixers, big impellers in them. Many of them, the more modern ones, have impellers. So it's not a sludge that's in the digester. It's actually it is a liquid slurry. So we've basically taken the cotton portion and turned it into a liquid slurry that would be very compatible with the inside of a modern anaerobic digester environment.
Ah, gotcha. Okay, thank you for getting into that. And then we have another one. Could you explain your cellulose enzymes mix? It seems like the crystallinity is a limiting factor.
Another wonderful question. The cellulose polymer and the cotton fibers are really amazing. They're just incredible materials. Um, the way nature produces them is also incredible, and I don't have time for that in this presentation. Maybe there will be another occasion. Um, it turns out that one of the reasons why cotton fibers are such great high performance materials, you could say like off the shelf or off the plant. I mean, the plant is making these beautiful fibers for us. We don't have to extrude them. They're just grown on the plant in this way. Those fibers have regions that are highly crystalline, very tightly packed, very organized, held together with hydrogen bonds. That's what stabilizes the crystallites in cellulose. And then the kind of the area between the crystallites is more disordered regions. People tend to refer that as the amorphous or disordered regions. So the idea when you find out how cotton degrades, actually we learned how cotton degrades through this work. We find out that really the enzymes are attacking those disordered regions and leaving the highly crystalline portions behind. If you treated it long enough with enough fresh cellulases over time and removed the glucose, because glucose is actually it causes product inhibition. So as long as you remove the glucose product and keep treating that crystalline cellulose with enzymes, you would eventually degrade the entire thing to glucose. So that can happen, but it would take a lot more time and effort. Um, so we were looking at this as a way to quickly separate the cotton portion from the polyester portion. That was the driver. But if you wanted to demolish it all the way to glucose, you could. And the other part of that question was, what are the cellulases in there?
Mhm.
We used cellulases that are the most aggressive cellulases that are commercially available today were actually developed for biomass conversion to biofuels. And they are all based on a chassis from what's called the Trichoderma ricei microorganism. So there are a number of suppliers of enzymes that are aggressive, and they tend to be multi-component enzymes. So they'll have a mixture of what are called endoglucanases that chop in the middle of the polymer, exoglucanases that chop off from the ends of the polymer chains, and beta-glucosidases that take the small fragments, dimers, and chop them into glucose. So you do need the kind of the whole mixture if you want to go all the way to glucose.
Okay, thank you. And then this is an interesting one. Have you ever tested the effect of natural dyes like indigo on enzyme activity?
Thank you for that question. We have indeed. And indigo is, yeah, denim jeans. Actually, denim jeans, I think, could be actually a really great first pilot test for this kind of concept. And the reason is because if you're looking at classic 100% cotton denim jeans dyed with indigo, indigo it's actually a pigment. So it's kind of trapped as little physical particles deep inside the fiber structure. So unlike those reactive dyes, the indigo does not form any durable bond to the cotton fiber itself. So if you kind of imagine you've got this like indigo particle here and it's sort of wrapped around in cotton, well, if you chop all the cotton away, then you have your indigo particle. And like by filtration, in principle, you could separate the indigo. You could chop down the cotton, separate the indigo, and actually recycle the indigo pigment and reuse that. So it could have a really interesting dye recycling opportunity there.
Oh, that's great because I've always heard how difficult denim is to work with. So that's nice to hear.
Yeah, we get wonderful. We call it denim juice. It's a wonderful blue. It's like a smoothie. So I don't know if you'd want to drink it or not, but it sure is pretty.
I bet. And then I have one, and of course, PFAS has entered the conversation. How do dyes, chemical treatments, and textiles impact the eventual PFAS volumes in digestate?
Okay, well, so first of all, the enzymes can't do anything directly about the PFAS.
Yes.
The only place where I would say maybe biotechnology has a chance there is possibly to use um, more generically the idea that proteins are capable of selectively binding things. So, but you'd have to go to somebody else for that. I have no expertise in how to make those kind of materials. So maybe you could use some biotech methods to help selectively tag or bind with PFAS compounds. But it's a generic and general problem. I think maybe whatever tactics could be used to either induce precipitation or filtration or binding of the PFAS during a process like what we just described um, could be useful. In other words, you could basically maybe kind of deploy that kind of technology at the same time as doing this enzymatic fiber separation. We haven't explicitly tested to see if the PFAS would interfere with the enzymatic treatment. um, I kind of don't think so. That's my guess. I'd love to try it. And not sure about what it's going to do to the anaerobic digester. The PFAS really kind of, they're so inert. They kind of like sort of don't interact with things, but obviously it would be worth testing. I have lost audio. Are you there?
I'm here. I'm sorry, you couldn't hear me. What challenges might arise in scaling this process, Sonia?
So thank you. The scaling part is interesting. So first of all, we need a piece of equipment like a big belly washer, some kind of equipment that would have a tumbling action. Of course, you want to do that with the lowest energy cost you can. So something like a big front load washing machine with screens in it is sort of what we are envisioning would be useful there. In essence, a kind of like turning this into a continuous or semi-continuous type of process that you could keep feeding material into. The mechanical action is really critical for helping the enzyme. The other thing is the washing and the separation steps. So those are really like engineering kind of questions. What I really want to re-emphasize is remember our process conditions for the enzymatic treatment. pH 5, which is very mild pH, 50 degrees. 50 degrees helps speed up the enzymes, help them work better. 50 degrees Celsius, I'm talking about. 50 degrees Celsius is also practical because um, microorganisms in general don't like to work at 50 degrees. So you could help prevent contamination from your kind of glucose syrup by working at a little bit elevated temperature. And I had a third thing I wanted to say there, the washing step. Certainly, if you want to recover the polyester fiber, washing is going to be important. But then here's maybe a question to pose to the polyester recycling experts. Potentially, if recycled polyester also has like little bits and pieces in it, once you melt the polyester, you're going to run it through some kind of filter anyway. So as long as you don't have so much contamination in there that you're that your liquid polyester spin dope filters wouldn't be able to take it out. I think that could be handled also kind of from an engineering perspective.
Okay, thank you. And we're getting a lot of questions about materials, and leather is thrown in here. Are the items like secondhand bags made of textile mixed with leather classified as textiles or leather, depending on which material is more dominant?
Wow, I don't really know how to answer. That sounds like a little bit of at the very beginning of my talk, I said it was really hard to get numbers globally on what really constitutes a textile waste.
Uh-huh.
I actually have been drilling into some of the United Nations kind of global reports on materials and waste. The United States actually distinguishes between leather waste and textile waste. Other countries do not distinguish, so it's actually really hard to know. It kind of depends on where you are, whether that's even tracked, or
Hmm.
and I would say the reporting is pretty lacking there. That needs to be improved.
That makes sense. And this is an interesting one about reuse. Is there any opportunity to reuse the residual dyes or the residual cotton fiber that we get in slurry?
So yes, reuse of dyes I think is realistic, certainly in the case of indigo, because in the indigo example, um, the indigo being a pigment, I mean, basically it's kind of like blue dirt on your clothes. So as long as you recover your blue dirt, you'll still be able to reprocess that indigo in the same way that you would normally apply it to denim, and that there's some chemistry involved in that process that we won't go into here. But in principle, indigo is a really good one that would have a lot of recycle potential. Um, the reactive dyes. Kind of less obvious how exactly you would do the recycle there, because once that fiber reactive portion of the dye has reacted with cellulose, that chemistry is gone. But what I will point out is we made blue slurries and we made red slurries. We made blue and red particles. Those are now, in my frame of thinking, those we've just turned them into pigments. So maybe you could actually just mill it and put it into a print paste and not use it as necessarily a reactive dye, but just use it as a brightly colored pigment instead.
Oh, nice. And then Sonia, do you think there's anything policy-wise that would interfere with this actually being scalable and used in municipalities eventually?
I am so not a policy person. I mean, policy. Wow, I mean, I think to me, I'm looking at like the what EREF, like the at the basis of what EREF is trying to do, which is trying to have new alternative solid waste treatments encouraged and deployed in the United States. So any kind of policy that would favor using anaerobic digestion, any kind of policy that would help the logistics of collecting and sorting materials and bringing them to facilities that could handle and process them, those would all be great policies. You know, kind of open the box on what kind of waste treatment options there could be for textile materials and other materials in general.
Thank you. That makes sense. And it's such an open area that we're all watching. So
Yeah. Oh.
um, here's another one. Can we use this dyed cotton fiber of slurry to mix up with virgin cotton to produce fiber dyed yarn?
Um, I'm not 100% sure I understand the question exactly. I think um, in the specific cases that I showed using the fiber reactive dyes, once it's been degraded down to kind of these small particles as slurry materials, you'd kind of have to handle them more like a pigment than as a classic dye stuff. And so pigments would normally be applied in like a printing operation. So you could definitely do that. The other possible aspect to the question would be there fibers like rayon are produced in solution processes where in principle you could add those colored ingredients into a rayon or a lyocell process. I don't know how the colorants themselves would tolerate the solution processing conditions, because there's some different kind of aggressive chemistry going on there. So the dye might not like that so much, but it could be tried.
Okay, thank you. And then I know we did our survey just to see how many of us watching today throw out clothing and shoes, and 37% of us said never, 63% say we have. So with that in mind, do you think this type of process could keep up the scale of textile waste, since we are throwing away so much?
So I think my comment there is that we need a variety of treatment processes to deal with all the complexity of the types of wastes that we generate to have the lifestyles we want to do. What I would really encourage your audience to do is to be thoughtful. Read your labels when you're buying clothes. Maybe maybe that's what I would ask, is from this webinar, just to be a little bit more informed about the types of fibers that are used in clothing, and what they have to, what needs to be considered in order to recycle them, and be able to bring them back into the front end of the process pipeline.
Thank you. That makes sense too. You've shared so much. Um, we really appreciate it. Thank you for your time and really sharing so much. And we look forward to you scaling this and seeing what's next for you.
Yeah, thank you very much, Liz. I really appreciate you and the support of the ERF. That was a very exciting project for us, and we're really pleased with the findings.
Excellent. And thank you to our audience for joining us today. And we hope to see you at our next science session on anaerobic co-digestion on September 18th. Save the date. And you'll also be getting an email from EREF through GoToWebinar soon. So if you could complete that brief survey, and then you can download your certificate. And we really do use those comments and your feedback, and they help us improve future science sessions. So thank you, everyone, especially you, Sonja, and enjoy the rest of your day.
Great, thank you.